Hello class 11th. Today we will discuss Archaebacteria and Eubacteria. Okay, this Archaebacteria and Eubacteria comes under kingdom monera, and general characteristics of kingdom monera that's I already taught you in the previous video. So today we will discuss about Archaebacteria and Eubacteria. Okay, this topic is very important for your NEET exam also. So if you are preparing for yourself for the NEET exam, so please focus this topic very clearly. We will try to know about Archaebacteria and Eubacteria by looking some points, okay, and that make the difference between these two. Okay, so let's start the topic. First of all, we will focus these both Archaebacteria and Eubacteria by their name, by their habitat where they found, okay, by the composition of cell wall, by the composition of cell membrane and plasma membrane, and by their nutrition habits. Okay, so let's start the study. First of all, we will discuss about Archaebacteria. Archaebacteria is formed by two words, archae and bacteria. So archae means ancient. Ancient means old. So Archaebacteria are ancient bacteria. That means primitive prokaryotic bacteria. Okay, and these are also called the living fossils. Now question comes, why these are called living fossils? So because from the beginning to till now, there is no change, no modification in their structure across the time. Okay, so that's why these are called living fossils. Okay, if you will see the habitat of Archaebacteria, so Archaebacteria found in a very harsh conditions. Okay, very critical conditions they found. Like they found in high salty area, they found in very high temperature area, and they found in highly acidic area. Okay, so the Archaebacteria's habitat is extreme conditions area. Okay, according to the basis of habitats, we can divide the Archaebacteria into three types. First is methanogens. As by name, methanogens formed by methane names. So methane loving bacteria. So methanogens are called the methane loving bacteria, and these are found in a marshy area. Okay, in marshy area the amount of the methane is very high. So that's why, because these are the methane loving bacteria, so these found there. Okay, if you will see the another type, so this is Halophiles. Halo means salt, salty. So these are the salt loving bacteria. Okay, Halophiles are what? The salt loving bacteria, and these found in extreme saline conditions. Okay, where the amount of salt is very high. So there we can see the Halophiles bacteria. Okay, salt loving bacteria. Next is thermoacidophiles. Means the thermoacidophiles, therm means heat and acid is the another acidic compound. Okay, so thermoacidophiles found where the temperature should be high and it should be very acidic environment. Okay, so there we can see the thermoacidophile. So got all three types. Okay, now if we will see the Eubacteria, so Eubacteria found everywhere, mostly in a familiar conditions. Okay, so Archaebacteria only they found in a very harsh conditions. Okay, very extreme conditions, right? So that makes the difference between the habitat between the Archaebacteria and Eubacteria. Okay, now we will see cell wall. Cell wall of the Archaebacteria and Eubacteria is different. Here we will discuss the cell wall of Eubacteria. In the cell wall of Eubacteria's cell wall is made up of peptidoglycan. And the peptidoglycan cell wall having two parts, peptide part and glycan part. Okay, so peptide part of the peptidoglycan is made up of 4 amino acids. Okay, these 4 amino acids are L-alanine, D-alanine, D-glutamic acid and L-lysine. Please note down these 4 amino acids, okay, which synthesis the peptide part of peptidoglycan of the cell wall of Eubacteria. Okay, again I am repeating, these 4 amino acids are L-alanine, D-alanine, D-glutamic acid and L-lysine. So these 4 amino acid synthesis peptide part of the cell wall of Eubacteria. Okay, the another part of peptidoglycan is glycan parts, and this glycan part is made up of 2 very important compound. Okay, first I called NAM and second I called NAG. Okay, NAM, NAM and NAG, NAG. Now write the full form of these 2 compound. NAM, NAM, having the full form N-acetyl muramic acid. N-acetyl muramic acid. Okay, where NAG having the full form N-acetyl glucosamine. Okay, written. So these 2 compound are very important, which just synthesis the glycan part. So if you will see the proper composition of the peptidoglycan of cell wall of eukaryotic bacteria, so it is made up with the 4 amino acid and NAM and NAG part of peptide and glycan part. Okay. In another hand, if you will see the cell wall composition of Archaebacteria, so in Archaebacteria cell wall is also made up of peptidoglycan, but in peptide part of Archaebacteria, the D amino acids are absent. Both D-alanine and D-glutamic acid. These both amino acids are absent in the peptide part of Archaebacteria. Okay, and if we will see the glycan part of the Archaebacteria, so Archaebacteria glycan part, it doesn't have the NAM part. NAM part is absent in the glycan part of Archaebacteria. So the total composition of peptidoglycan and the cell wall of Archaebacteria, so in the cell wall of Archaebacteria, the 2 amino acid, D group amino acid, and in glycan part NAM is absent. Okay, so that makes the difference between the cell wall of Archaebacteria and Eubacteria. So that's why Archaebacteria can survive in very extreme conditions because of this ability only. Right? Now we will discuss about the plasma membrane and the cell membrane of Eubacteria and Archaebacteria. So first we will study about the cell membrane of Eubacteria. Right? Cell membrane of Eubacteria is made up of phospholipid bilayer. Both layers are present there. Okay, in the cell membrane. So that's why this is called phospholipid bilayer. Bilayer means 2 layers are there. Okay. In case of Archaebacteria, the cell wall is the plasma membrane or the cell membrane is also made up of same as eukaryotes, means phospholipid, but it is in monolayer. The single layer is there. In case of Eubacteria, it is bilayer of phospholipid, but in Archaebacteria, it is monolayer. That means single layer of phospholipid. So this is the difference between the Archaebacteria and Eubacteria plasma membrane. So here you have seen the difference in the Archaebacteria and Eubacteria cell wall and plasma membrane or cell membrane. So these 2 differences can make the Archaebacteria different from Eubacteria, and that's why because of these 2 differences only the Archaebacteria can survive in extreme condition. Okay, right. Now we will see the nutritional habit. Okay, and this nutritional habit we will discuss first of all with the Archaebacteria. So in nutrition we will see the nutritional habit of the three types of Archaebacteria. Methanogens, halophil and thermoacidophil, they having their different nutritional habit. Okay. So first of all we will discuss about the methanogens. As by name methane, the methanogens are called the methane loving bacteria. So of course these will found where the methane is present. So these methanogens basically found in the gut of animal, okay, where the methane is present. If we are talking about the cow, so in the gut of cow we can see methanogens also. These methanogens having the ability to convert the cellulose compound into methane. Okay, so if you will see the cow dung, so cow dung having the ability to produce methane gas, and that you will see in biogas plant. Okay, so what happened there? We use cow dung and by that methane gas generate, and this methane we can use for the kitchen flame purpose or kitchen cooking purpose, right? So here this cellulose is converting into methane by the action of methanogens. What these methanogens do, these complete their oxidation process there, and these convert the cellulose into the methane by the fermentation process in biogas plant. Okay, and by that when they are just converting the cellulose into the methane gas, they produce ATP, and these ATP they use for the formation of food, right? So basically these are making the food for themselves. So that's why these methanogens are chemoautotrophic. These using the chemical to synthesize their own food. So that's why these are called chemoautotrophic bacteria. Okay, methanogens. Got it now? Now second is about halophils. Halophils are chemoheterotrophic. These depend on others. Heterotrophic means depend on others. Although they having their chemical and they got sunlight there, proper sunlight, and this sunlight help the halophils to produce ATP. But this ATP they can't use for the synthesis of food. So that's why, because they can't make their own food by these ATP. So that's why these are depend on others for the other compound for the food. Okay, that's why these are called chemoheterotrophic bacteria. The halophils are chemoheterotrophic bacteria. Okay, now we will discuss about the thermoacidophils. These thermoacidophils always found in the hot spot area where hot springs are present. Okay, so in hot spot area you can see sulfur is always present there. Okay, and thermoacidophils convert sulfur into sulfuric acid. Got it? So when these are converting the sulfur into sulfuric acid, so by this process they got always ATP. Okay, and this ATP they use for the synthesis of their own food, right? So because here these are thus using the chemical processes for the synthesis of their food, so that's why the thermoacidophils are also known as chemoautotrophic. Okay, so thermoacidophils are what? Chemoautotrophic, and they can synthesize their own food. So in eukaryotic bacteria, the eukaryotic bacteria having the variation in nutrition mode. These may be heterotrophic. Most of all these are heterotrophic. Some are autotrophic, some are parasite, and some are saprotrophic. So variation there in the nutrition mode in eukaryotic bacteria. In eukaryotic bacteria, the another type of bacteria is present, and that is called the Mycoplasma. Please focus this Mycoplasma. Why I am teaching you this Mycoplasma separately? Because this Mycoplasma is a very different in Eubacteria. Okay, it is Eubacteria, but this Mycoplasma, it don't have this cell wall. Okay, this Mycoplasma don't have cell wall. And because it don't have the cell wall, so that's why it can change its structure and shape. Okay, so because the Mycoplasma can change its structure and shape, so that's why this is called the monera. pleuromorphic. The Mycoplasma bacteria is called pleuromorphic bacteria. Why this is called the pleuromorphic bacteria? Because it can change its structure and shape. And how it can change its structure and its shape? Because it don't have the cell wall. Okay. Now, this Mycoplasma can produce the disease in the plant as well as in animal. In plant, the Mycoplasma can produce the which is broom disease. Okay, which is broom disease. Mycoplasma can produce in plants, and plant structure can also be different at that time. Okay, some bushy like structure it can synthesis there. So one side it can change its own structure, and another side it is producing the disease like it, which changing the structure, a little bit structure of plant also, which is called broom. So that's why it is called the jokers of the plant kingdom. The Mycoplasma is called the jokers of the plant kingdom. Got it? In another hand, in animal, this Mycoplasma can also produce disease. Okay, and that is called the pleuropneumonia. Okay, this pleuropneumonia disease the Mycoplasma can produce in animal. Okay, and because of the name of disease pleuropneumonia, this Mycoplasma also called PPLO, the very important, okay, is producing the disease pleuropneumonia in the animal. That's why this is called PPLO. And what is the full form of this PPLO? The pleuropneumonia like organism. Pleuropneumonia like organism is the full form of PPLO. Okay, and why the Mycoplasma is PPLO? Because it can produce the pleuropneumonia disease in animal. Okay, so children, that's all for today, and that was the information of Archaebacteria and Eubacteria. Now you got the difference between these two. Okay, so this is all for today. During this lockdown time, stay at home, save your and your family life. God bless all of you.